Hi, everyone. I hope you enjoyed the talks and workshops so far. I'm Nasim Jamali. I was a postdoc in the imaging platform of the Broad Institute in Dr. Beth Cimini's group until recently, and I joined DuPont Therapeutics as a data scientist a couple of months ago. Today, I'm sharing with you the results of a BioImage Analysis Survey that was conducted in 2020 through the NIH-funded Center for Open Bioimage Analysis. And we published the results last year in the Journal of Biological Imaging. The main goal of this survey was to find the software and training material needs of the bioimaging community and provide resource for tools and software developers in this field. I would like to thank the biology and microscopy community for providing answers to this survey, the co-authors and members of Cimini, Carpenter, Singh, and Eliceiri labs for their input on the questionnaire and assessment of needs in the bioimaging community. The 484 survey participants were mainly from North America and Europe, and mostly trainees, consist of postdoctoral fellows, graduate and undergraduate students, followed by principal investigators, facility staff, scientists, and image analysts. And majority of research area were in biological sciences, biochemistry, followed by physics and computer science. We asked several questions along a linear scale, a scale of 1 to 7 scores. And in response to the type of work question, survey participants described their work as mainly balanced between hands-on imaging and lab work, a score of 1, and image analysis and data analysis, a score of 7. We also asked them about their computational skill level. And in this figure, broken down into job categories, we can see that the computational skill level somewhat follows the roles. And as the roles become more or goes more toward image and data analysis, the computational skill level increases as well. 43% of survey participants were trainees. So we wanted to know the difference between trainee and non-trainees in area of work and research, level of computational skills, and comfort in developing new computational skills, which was another question asked in the survey. As you can see in this figure, in both trainee and non-trainee groups, the majority of participants were from the balanced group between hands-on imaging and analysis. And trainees didn't report themselves as having the highest level of computational skills, but overall the level of comfort in developing new computational skills remain pretty similar between trainee and non-trainee groups. We then wanted to know the type of images mostly analyzed by the community and their modality and technique. As we can see, bright field, DIC, and phase, followed by fluorescent two-dimensional images from cells and organisms, were the most commonly analyzed images. The type of tools the community uses to analyze their images are mostly open source point and click tools such as ImageJ or Fiji, Cell Profiler, and IC, followed by computational libraries and scripts such as Scikit-Image and MATLAB libraries. And they mainly prefer to sit down with a tool they know and search the web as scientific literature to find solutions for their image analysis problems. To better understand the needs of bioimaging community and what areas are needing focus for the tool developers, we asked about the type of images, the type of image analysis problems that the community wished that they had better solutions via open-ended question. And by parsing the responses with Python scripts, we found that object segmentation in general, and especially in 3D, along with tracking and tissue or histology analysis, were the highest ranking responses. We then wanted the community to provide suggestions and feedback for the developers and users through open-ended questions. And they encouraged developers to improve documentations, user interface, plugins and packages, as well as providing easy to follow workshops and tutorials. There were also suggestions such as prize offerings for best tool instructions. The community asked users to seek out existing tutorials and workshops and learn the tools and basics of image analysis, follow the best practice guidelines for imaging, and ask their image analysis question on a central location, such as the Scientific Community Image Forum or forum.image.sc. This could make it easier for tool creators to provide answers. There were also suggestions for frequent reminders for users to cite analysis tools in publications. They encouraged users to start image analysis when performing pilot imaging and condition optimization experiments rather than waiting until the completion of larger scale experiments. Participants also suggested that principal investigators encourage their trainees to take image analysis courses and try new image analysis tools. We further categorize these responses based on type of work, and you can see those details in the paper. And to further understand the community's need about the method of learning in regard to the topics related to image analysis educational material, we asked participants to rate their interest about the different method and lengths of learning. Written and video tutorial followed by webinars and office hour showed the highest interest level among the different type of learning methods. These results remain somewhat similar when we further broke down the results between the trainee and non-trainee groups, and based on level of computational skills and comfort in developing new computational skills, which again, you can see those figures in the paper. In general, the community thought what makes a specific resources beneficial are basics and background information, step-by-step and hands-on approaches, followed by immediate feedback and having access to an expert during live sessions. And the community prefers to be notified about the events and trainings through mailing list and the forum, followed by Twitter and their local microscopy facility. To just mention a few points that we as a community could focus on, we want to think about how can we reach a larger community, the biologists and groups least exposed to the image analysis and available resources. One of the ways that Center for Open Bioimage Analysis is hoping to reach those groups is through increased collaboration with instructors to develop the image analysis sections of their trainings. Community also asked the field-specific scientific meetings and conferences to offer image analysis workshop and inform their members about the available resources. And in the currently available resources and forums, perhaps expanding the existing templates for common questions could make it easier for users to know which information developers and experts most likely need in order to answer their questions. And a central location for available resources along with a short description of them could help a new user to choose the best tool for their image analysis needs. And we are always looking for your feedback and suggestions, so please reach out. And thank you for listening, and I'd be happy to take any questions.